Thank you for joining us today for BD FACSMatter Ask the Expert webinar on understanding COVID-19 and immune responses by two guest speakers and moderated by Bob Balderas. We are excited to share this program with you. Before we start the program, I would like to introduce Bob, who would be doing the introduction for our guest speakers. It is an honor and a pleasure to introduce Bob, who is our resident subject matter expert. Bob is a Vice President of Biological Sciences at BD Biosciences. He is a distinguished fellow with over 40 years of leadership experience in both academia and industry. Bob serves on the editorial board and of various eminent scientific journals, and also is a Dean Counsel at UCSD School of Biological Sciences. He has over 75 scientific publications and is recognized all over the world as a visionary that helps create academic and industry partnerships for advancement of science. He has been honored with two prestigious BD Howey Awards. Bob has a passion for sharing best practices in areas of immunology, high-dimensional biology, and flow cytometry. He has participated in worldwide educational seminars and tours for the last 20 plus years, and we are excited to have him moderate this session for us. Before I hand it over to Bob, I would like to go over a few housekeeping items. If you have any questions during the webcast, please submit them through the Q&A widget of your console. Our scientists will be answering them on the back end. And after the presentation, we will have a live Q&A session with Bob and our guest speakers. For the best viewing experience, we recommend using a wired internet connection and closing any programs or browser sessions running in the background that could cause issues. Some network cause slides to advance more slowly than others, so we recommend logging off VPN. If your slides are behind, pushing F5 on your keyboard will refresh the page. You can find additional answers to some common technical issues located in the Help Engagement tool at the bottom of your screen. Lastly, a recording of this broadcast will be made available after the presentation. Look out for an email with additional details on where to access it. And with that, thanks once again for joining us, and I hand it over to Bob. Bob, the floor is yours.
Thank you, Manisha. I really appreciate the introduction. Well, certainly today I welcome everyone, and it's always an esteemed honor to have an opportunity to introduce, distinguished scientists for today's program. And today, the focus is going to be on understanding COVID-19 and immune responses. Our first speaker is Dr. Deepak Kaushal, who is currently the director of the Southwest National Primate Research Center and professor of the Host Pathogen Interaction Program at the Texas Biomedical Research Institute. Dr. Kaushal holds a PhD in biochemistry and microbiology from the University of Delhi, and is a member of many, many different types of societies, including the Infectious Disease Society of America, the American Society of Biochemistry and Molecular Biology, also on the Bill and Melinda Gates Foundation for collaborations for TB vaccine discovery, as well as on the working group for the non-human primate models. In addition to that, Dr. Kaushal is also responsible and works hard on the AIDS clinical trials group. Now, Dr. Kaushal has focused most of his career on the eradication and the understanding of TB and the synergy with HIV. But today, the title of his talk will be TB and COVID-19, connecting the dots between both diseases. Now, our second speaker is Dr. Dhiraj Singh. And Dhiraj is a staff scientist also at the Texas Biomedical Research Institute and the Southwest National Primate Research Center. He holds a PhD in immunology from the International Center for Genetic Engineering and Biotechnology, and also holds a Master of Science in Biotechnology. Dr. Singh's research is focused on T-cell immunity. in TB and TB-SIV co-infection and its applications for developing novel immunotherapies and vaccines. Today, though, he too is utilizing his experience for assessing therapeutic and protective interventions against COVID-19. The title of his talk will be Using Flow Cytometry as a Tool to Understand Immune Response. So our first speaker is Dr. Deepak Kaushal.
Thank you, everybody. And thank you, Manisha and BD, for inviting me to give this talk. Today, I'll be talking about TB and COVID-19, connecting the dots. I'm Deepak Kaushal. I'm the director of Southwest National Primate Research Center, one of the 7 such NPRCs in the country that are primate facilities with expertise specifically supported by the NIH. And the SNPRC is within the host body of Texas Biomedical Research Institute, a not-for-profit research institute that is based in San Antonio. So I'm very happy to talk about our work with you. For almost all my life, I've studied TB as a researcher. Tuberculosis is a disease of infectious disease of high magnitude that leads to between 1 to 2 million deaths every year still today, and is therefore very worthwhile studying. And for the past 15 years, I've been focusing on the study of TB using non-human primate models. This was facilitated in 2006 when I joined Tulane National Primate Research Center as my first faculty position. So we study TB in non-human primates primarily because the pathology, the granulomas that are the hallmark of tuberculosis, are recapitulated in non-human primates, for example, rhesus or cynomolgus macaques, as seen here in this slide. So when you look at human granulomas or granulomas that are formed in macaques, you see a very high level of organization with internal necrosis in a region that is hypoxic, followed by defined layers of macrophages, lymphocytes, and fibrotic cells. And these are really very important concepts. This level of organization has a profound impact on the bacteria, as well as host responses that lead to the interplay of either the control or the progression, either the control of infection or the progression of disease. And unfortunately, it's very hard to mimic this level of granuloma organization and the downstream control versus progression phenotype in rodent models where granulomas are not very organized. And this is primarily the reason why I came into macaque models. Many before me, like Dr. Joanne Flynn in Pittsburgh, had blazed the trail to develop these models, and many before her had shown that these animals are very good models to study several specific aspects of AIDS infection and HIV progression. However, in early this year, about 2 years ago, actually, I moved to San Antonio from Tulane, and we built a brand new large monkey ABSL3 facility, and we've since actually expanded it and almost doubled our capacity. So for much of 2019, we were unable to do any experiments as our facility was being constructed. And late last year, we were able to have our facility ready, and then Dheeraj did one experiment in that building. And soon after that, COVID started taking hold. And at that point of time, we had a huge ABSL3 facility, and we had non-human primates in our center. And I had a cadre of postdoctoral research scientists and technicians and grad students that was specifically trained to use various aspects of this ABSL3 facility, infect and treat non-human primates, take samples from them, and then study those samples, especially with respect to their immune responses. So we decided, upon instruction from the National Institutes of Health, to develop models of non-human primates species with SARS-CoV-2 infection and COVID-19 disease. So we chose 3 different species to initially study. These were the Indian origin, specific pathogen-free rhesus macaques, African origin, mostly specific pathogen-free baboons, and then New World origin, mostly Brazilian origin, marmosets. So 3 different species in size and where they come from, but non-human primates nonetheless. And there was rationale for choosing either species. The Indian rhesus macaque is the model of choice for doing infectious disease studies in the non-human primate world. And baboons in particular, have some history of infectious disease work being done, but more importantly have been used as models for cardiovascular disease and metabolic diseases like diabetes, which have been thought from early on to be confounding factors and comorbidities for COVID-19 progression. And so it was important to study, in my opinion, baboons. And then marmosets were really useful for developing models of Zika. And so we thought that we should at least give a try with respect to marmosets also. We were helped by the fact that all 3 of these species we have breeding colonies for in the SNPRC. And then we took animals from each species that were young and old, mimicking adolescent to very young adult human beings to maybe septuagenarian, octogenarian human beings, and we decided to study SARS-CoV-2 infection as a function of age. So the first thing that we did was we decided to do a pilot experiment in which to try and develop the model. So we took a small number of rhesus macaques, 4 of them, and we decided to expose them to SARS-CoV-2 through multiple routes, ocularly, intranasally, and intratracheally, giving 1 to 10 to the power 6 of the Washington strain, the Washington state strain, the Seattle strain virus, which was highly characterized, obtained from BEI resources, and then propagated to low passage in our facility by our virologist, Ricardo Carian. And we did a very short pilot study where we exposed these 4 animals to the virus, and we studied disease correlates. And what we found was, as shown in the slide, that there were signs of acute infection and inflammation due to infection. So if you see the levels of serum C-reactive protein were significantly induced a day after infection and stayed significantly high throughout the course of this study, which was a 3-day study. And then if you look at levels of albumin, they dropped down significantly on a daily basis. And the same was true for hemoglobin, thus indicating systemic inflammation as a result of viral infection and maybe viral anemia. These animals also exhibited high levels of viral load in various compartments. On the left, I'm showing you viral loads in the bronchoalveolar lavage. And you can see that there is about 4 logs more virus at day 1 as compared to day 0, and about 3 to 4 logs more virus at day 2 as compared to day 1, thus showing that there is clear replication of this virus, at least in the lower respiratory tract. And then by day 3, you see a control of infection in the lower respiratory tract. And in the upper respiratory tract, in the nose, in the nasopharyngeal swabs, you see there is maintenance of virus, but perhaps because this compartment is less accessed by immune cells as compared to the lower respiratory tract, you don't see rapid clearance of the virus from the nasopharyngeal swabs. And then finally in the throat, in the bucopharyngeal swabs, you see a similar story. You see some level of replication and persistence of virus in this compartment over 3 days, but clearly evidence of replication. And then when we necropsied these animals at 3 days and we looked at every lobe of the lung, each one of the 7 lobes of the lung, and we were able to detect high levels of viral RNA in the lungs of these animals. This is a genomic viral assay. Each animal is shown with a different color. Each of the 4 animals, we were able to show about 6 logs of viral RNA being present in the lungs of these animals. And when we ran a subgenomic assay, which is a correlate for replicating viruses specific to E, we saw about 2 logs lower levels, about 4 to 5 logs of subgenomic RNA, but again indicating replicating virus in the lungs of these animals. And then these genomic and subgenomic levels were highly characterized. We then looked radiologically into these animals that had been exposed to SARS-CoV-2. And by day 3, our X-rays clearly showed evidence of ground glass opacities and pneumonia. However, actually you can look at, you can look at these X-rays and score them. And what we found was, and this was, by the way, scored by a board-certified veterinary radiologist that is not on site at our center, but is double-blinded and off-site, and she or he just receives data as packages with no indication of what the experiment is or what the specific animals are, and so they score this completely blindly. And over the course of time, between day 0 and day 3, you could see that our radiologist scored our animals as increasing score of pneumonia and ground glass opacities in X-rays. However, we were able to also subject these animals, the rhesus macaques at least, to high-resolution CT scanning. And this was, in my opinion, a major advance that we had over some of the other groups that were trying to do this. And so we can CT scan these animals from 3 different angles. On the left, I'm showing you from the top, and in the middle, I'm showing you CT scans from the front, and then finally on the right, I'm showing you CT scans beamed from the side. And again, as shown in this example here, the same animal imaged in the same position at the same time of the day over the course of day 0, day 1, and day 2. And you can very clearly see development of ground glass opacities marking pneumonia in all of these animals. In some cases, the ground glass opacities appeared at day 1 and stayed at day 2 and day 3. In other cases, they appeared either at day 2 or day 3. And in some cases, they resolved by day 3, but many of these were maintained. And again, when these were scored by our radiologists, we saw a huge bump in our scores between day 0 and day 1, and then maintenance at day 2, and then some lowering of the score at day 3. And then we were able to use CT scans to also look at pneumonia. So one of the things you can do using these CT scans is in a higher resolution, look at how the cells are arranged in the lung. And are the cells arranged in a hyperdense manner, or are they arranged in a normal manner? So normally you would expect cells to be arranged in single cell formations in the lung. And when you have pneumonia, interstitial or alveolar pneumonia, then you get too many cells infiltrating the same area, and then this causes fluid. So we use hyperdensity as a correlate for pneumonia. So you can see here on the top, pseudo-colored, of course, but at day 0, there's some level of hyperdensity, and then most of the lung is normal. At day 2, in one of these animals, you see evidence of greater hyperdensity or greater levels of pneumonia, as you can see, particularly from the top and also from the front. So clearly there was evidence of greater pneumonia, and this could also be scored as a correlate of hyperdensity. And you see high levels of hyperdensity increasing progressively over the 3 days of infection. And then we were able to also look at cytokine expression profiles in the bronchoalveolar lavage. So some of the cytokines that we studied are pro-inflammatory cytokines and type 1 interferon level cytokines, as shown here. So if you look at IL-6 levels in the bronchoalveolar lavage of these animals, you see logarithmic or multi-logarithmic level increases from day 0 to day 1, and then further increases at day 2 or day 3. And the same is true, you can see here, for type 1 interferon molecule, interferon alpha, and many of the molecules that are downstream of type 1 interferon in signaling. So clearly, you have those cytokines that are important for the formation of COVID-19 disease and ARDS syndrome being expressed to high levels in these animals. And when we look at pathology, our pathologists were able to see lobe level discoloration as well as interstitial pneumonia marked here, with macrophages, lymphocytes, neutrophils, and basophils appearing, and levels of fibrosis and bald hyperplasia that are not seen in normal rhesus macaque lungs. Again, recapitulating various aspects of COVID-19 pathophysiology and pathology that is seen in some human cases in these non-human primates. What we found, however, was that SARS-CoV-2 infection was self-resolving in rhesus macaques. So we then, after we had established the basic model, we did a second study where we followed the virus and other attributes for a longer period of time. So we infected with the same dose, the same route, the same type of virus, the same strain of virus, but followed the animals for 2 weeks, and then we necropsied them. And as shown on the top for rhesus macaques, the peak viremia in bronchoalveolar lavage occurred at day 3, and after that the virus was efficiently controlled. In the nose, again, the peak was at day 3, and the virus was not efficiently controlled, but just persisted. And in buccopharyngeal and swabs and in other samples, the virus was also efficiently controlled. There was hardly any evidence of systemic spread of the virus. We didn't find virus in the blood or the urine, and in the rectal swab, as shown here in panel C, there was hardly any virus. We also infected baboons. Just like rhesus macaques, we infected baboons. And again, like rhesus macaques, we had 12 baboons, 6 of which were young, 6 of which were old. And we saw pretty much the same story in baboons, same levels of peaks of virus at the same time, day 3, and then after that, the virus declines. You see a little bit greater and more clearer decline of the virus here in baboons in the nose as well. And you see very low levels in the buccopharyngeal swabs, so not a whole lot of persistence. The only difference was that for some reason we saw huge levels of virus in the rectal washes of these animals as compared to rhesus macaques. So this is something that we are looking into now. It looks like that while rhesus macaque is a self-resolving infection, and so is in baboons, but for some reason there is more disseminated spread of the virus in the rectal swabs. And Luis Giavidoni from our center has actually shown the presence of virus, more presence of virus in the baboon gut as compared to rhesus macaque. So clearly this virus goes into the gut of baboons. So again, showing you that in different non-human primates, you can get more gut-based rectal type of disease that's been observed in some human beings, whereas in other animal species, you only get lung-based disease. Dheeraj is an outstanding cellular immunologist in my lab, and you'll hear from him in a moment. And he conducted high level of flow cytometry-based immune evaluation in all of these animals. And what he initially found was that that there was an extensive influx of myeloid-type cells in the lungs of animals that had been exposed, specifically rhesus macaques that had been exposed to SARS-CoV-2. So here I'm showing you data on top in black and white graphs, bar graphs, and split by age at the bottom, with older animals shown in red and younger animals shown in blue. But the results point to the same thing. If you look at panel A, which describes interstitial macrophages, or panel C, which describes neutrophils, or panel D, which describes plasmacytoid dendritic cells, you see an increase in those levels in the bronchoalveolar lavage. The recruitment to bronchoalveolar lavage is increased significantly at day 3 compared to day 0, which is a baseline sample. So at day 3, where we had the maximum amount of virus present in the lower lung, you also had the maximum amount of these cellular infiltrates coming in, probably explaining the results that we saw by pathology and by CT scans. But by day 9, these levels ameliorate, they normalize, again showing that this disease, this infection is self-limiting in rhesus macaques. And the only difference was alveolar macrophages, which we consider are 2 or 6 positive macrophages and are resident in nature. Their percentage slightly declined, while other percentages were increasing. This is probably because you're getting influx of other types of cells. The key thing was that there wasn't much difference between younger and older animals. We saw the same level of influx, myeloid influx and myeloid pneumonia being caused in both types of animals, independent of the function of age. And the influx of neutrophils and interstitial macrophages and PDCs was significant to highly significantly correlated with control of infection. These myeloid cells that appeared into the lung compartment as a result of SARS-CoV-2 infection also could be productively infected with SARS-CoV-2. We think so because we can at least detect SARS-CoV-2 proteins. In this particular case, nucleocapsid, but we've done this for both nucleocapsid and spike protein. So here you can see, for example, the presence of SARS-CoV-2 nucleocapsid protein in cells that are being marked with CD68 as macrophages and CD123 as plasmacytoid dendritic cells and CD66 ABCE as neutrophils, all of them carrying plenty of signal for SARS-CoV-2 specific proteins. And then Dheeraj also looked at dynamic changes to the T cells in the lung compartment, and what he found, as seen here, was again there was no difference as a function of age, but there was recruitment of T cells early on, again peaking at day 3 and then normalizing at day 9, both for CD3 and CD4 positive cells as shown here. And then these cells expressed high levels of activation and really energy markers, PD1 and LAG3, again suggesting that T cell responses in this model that are mounted early on to SARS-CoV-2 specific proteins are really critical for the control of this infection, and also suggesting perhaps that there is exhaustion of these cells eventually. And then Dheeraj very elegantly showed that there was ability of these T cells, CD4s and CD8s, to actually produce cytokines de novo. So here we are showing you data for IL-2, and clearly you can see over the course of time, IL-2 production increases on CD4 cells, as well as granzyme B production as shown here. So clearly these cells are able to produce cytokines. Surprisingly, however, when we did antigenic stimulation, so we looked at response to spike protein overlapping peptides as compared to a positive control PMA inomycin, Dheeraj was unable to see major increases in levels of cytokine production, again shown here for IL-2 and granzyme B, but we've tested this for other species, for other markers as well, clearly showing that there is some level of exhaustion that may be occurring in these T cells that have been recruited to the lungs of these rhesus macaques infected with SARS-CoV-2 infection. Surprisingly, however, while there were no differences in the types of T cells and myeloid cells that were recruited to the lungs of young versus old animals that had been infected with SARS-CoV-2, there were significant differences in the levels of SARS-CoV-2 specific antibody. So when we looked in the sera of these animals at necropsy 2 weeks after they had been infected, we found significantly lower levels of SARS-CoV-2 specific antibody in older animals as compared to younger animals. So clearly there are possibilities of immune response differences between young and old animals that could describe what happens in human beings. And it's possible if we do durability experiments of the type that we've planned but haven't done yet, if we look at 1 month or 1.5 months after infection, it's possible that we will see other differences in immune responses as well. I don't have slides here and I'm not showing you this data, but Tae-Young Lee from my lab, another experienced immunologist, has looked at NK cells and found very interesting functional differences in NK cells that are recruited to the lungs of these animals in old versus young animals. And these results, when taken together, have the propensity to perhaps explain antibody differences and functional differences in NK cells. So just to conclude this part of my talk, We developed models of SARS-CoV-2 infection in macaques and baboons. And I didn't talk about marmosets because we found that they weren't infected that much and there wasn't much progression. But at least macaques and baboons were infectable and showed various signs of SARS-CoV-2 infection ranging from clinical to radiological, immunological, and pathology. And we think that the rhesus macaque model is an ideal vaccine model because we're able to study immunology in that model, whereas the baboon model is an ideal therapeutics model for the space because we can do diabetes and other comorbidity studies in that model. So my lab has been very interested in studying responses to TB using lung-specific transcriptomics. And this work has been done in collaboration with my long-standing collaborator, Shabana Khader, who's a professor at WashU, Washington University in St. Louis, and with a very talented bioinformatician, Makedonka Mitreva, in the same institution. So in 2019, we published a Science Translational Medicine paper where we compared the lung-specific responses by whole genome transcription in mice, monkeys, and humans that had been infected with TB, mice and monkeys, of course, experimentally, and then humans using a cohort that had been studied, that had been obtained by and studied by our collaborator, Tom Skriba, in Cape Town, South Africa. So we decided to do similar studies in monkeys that had been infected with SARS-CoV-2. So 2 weeks after infection, the monkeys were euthanized, and we obtained their lungs and isolated RNA and subjected them to whole genome RNA sequencing. And we found some results that were unsurprising, as shown here, that genes that are involved in cytokine signaling were induced high levels in both the lungs of SARS-CoV-2 infected animals as well as the lungs of TB infected animals. Here I'm showing you red color as low expression and blue-green color as higher expression. So our uninfected animals have low expression of these genes, whereas of these induced by COVID-19 as well as by tuberculosis. And if you look at the specific pathways that these genes can be binned into, then they correspond to immune system, neutrophil degranulation, innate and adaptive immunity. So this was unsurprising to us. However, what was surprising to us was when we looked at genes that were not induced in TB, but were induced in COVID-19 as shown here. So, genes that are in red in TB-infected animals, but in blue-green, their expression is increasing in COVID-infected animals. Those genes could be binned into pathways like blood vessel morphogenesis and angiogenesis and vasculature. Again, hinting at the fact that while these rhesus macaques didn't have end-stage disease by which humans die of COVID-19, but they actually recapitulate some of the aspects of human COVID-19, where angiogenesis of the lung is a key correlate of death and of extensive disease. At least molecular markers for that could be studied. And so there, I think there's a very interesting interplay in how TB and COVID both lead to pro-inflammatory cytokine production, pneumonia, and lung damage primarily, but then the downstream progression of the two diseases may be different. So as I mentioned to you, we have a macaque model of SARS-CoV-2 infection and COVID-19 disease, which with measurable attributes. We can measure cytokines levels, we can measure cellular immunology, we can measure quantifiably pathology at the endpoint, and we can measure quantifiably CT scans-based pneumonia over the course of time longitudinally. So we decided to apply these to the study of various vaccines at our center. One of the prominent ones that we've studied is Pfizer. The Pfizer mRNA-based vaccines B1 and B2 have been in news, and the press releases and the news articles related to that, I think all of you have heard about, and we have very good data. Pfizer has very good data with their human trials. Before the human trials began, we actually did a non-human primate study at our center where rhesus macaques were infected with SARS-CoV-2 using the intranasal and the intratracheal route, and we gave the same 1 × 10⁶ PFU virus and these animals had been immunized with the mRNA viruses earlier. And then we looked at the presence of virus and immune responses. And here I'm showing you just some information about Pfizer B1 and B2 mRNA vaccines. The two different constructs are slightly different. One of them, both of them have lipid formulation. One of them is a modified mRNA that encodes the spike glycoprotein captured in a pre-fusion conformation for better immune responses, and that's the B2 virus. That's the B2 vaccine. And we studied both of them. And what Pfizer found was that both their vaccines, but in particular B2, was highly immunogenic in rhesus. So in panel A, I'm showing you spike protein specific antibodies being produced by these animals post vaccination, so 21 to 56 days after vaccination, and then after first vaccination. And then if you compare them to human convalescent sera shown in this graph, in this bar over here to the right, you can see that these animals are producing significantly more antibody that is specific for the virus spike protein as compared to human convalescent sera. And then when you look at true neutralization assays for the virus, again, you can see that plasma derived from these animals that have been vaccinated with SARS-CoV-2 specific vaccine B2 can actually generate much more neutralization. And then when we look at cellular immune responses, in this particular case, triple production of cytokines gamma, IL-2, and TNF-alpha from CD4 cells of these rhesus macaques, you see significant increase in triple production of these cytokines in vaccinated animals. Here we are showing you data from 2 different doses of the vaccine in green and in dark green as compared to unvaccinated animals. And the same is true for IL-4 production as well as gamma production from CD8 cells. So broad antibody as well as T cell responses. And then what we found was that Pfizer B2 was also fully efficacious in rhesus macaques. So here you can see data in bronchoalveolar lavage in panel A, and in the nose and in the mouth, in the throat, of animals that have been vaccinated and then challenged. And as you can see, the B2 virus vaccinated animals have very low levels of below threshold, really, of viral RNA being detected in the bronchoalveolar lavage, whereas the control animals have high levels of virus, as we would expect at day 3, the peak, as I showed you earlier in the model. And then our sentinel animals that were kept in the same room but were neither vaccinated nor infected, and they were right next to the animals that had been challenged, show no evidence of virus. So this was an important control to include. And again, in the nasal swab, the same story. The virus persists a little bit longer, as I've shown you earlier, primarily because this site is not as served by immune cells as the lower lung. But again, you see clearance of virus in the vaccinated animals. And then again, significant differences in the levels of virus, lower viral load, lower viral RNA load in animals that have been vaccinated with B2 as compared to unvaccinated animals. So clearly showing that this vaccine has the propensity to control viral replication and disease in rhesus macaques that have been vaccinated and that have been challenged with SARS-CoV-2. So I'll end here and acknowledge a whole bunch of people that made this work. Dheeraj Singh, a staff scientist in my lab who will be speaking after me, led this work along with Journey Cole, who's my lab manager, and several other members of my lab and other faculty members in Texas Biomed and core scientists in the SNPRC helped with this process. In particular, I want to acknowledge the help by Ricardo Carrion, who performed all the molecular virology, Luis Giavidoni, who performed all the cytokine analysis, and Shashank Ganatra in my lab, who did all the imaging. I also want to acknowledge Shabana Khadir's group in WASH U STL for RNA-seq, and support by the NIH, by BARDA and Regeneron, and by Pfizer. I want to acknowledge support by our group of veterinarians and veterinary pathologists led by Kathy Brasky in this instance, and BSL3 veterinary tech groups in SNPRC, as well as two individuals, our president of the Institute, Larry Schlesinger, and vice president for strategic planning, Corey Hallam, who were able to fundraise $3.5 million in a 2-week period towards the end of March, so that we could do these experiments in April, and allowing Dheeraj to write a paper by June, which has now been accepted. With this, I'd like to hand over the conversation to Dheeraj, who will be talking specifically about some of his use of flow data in this context.
Hello, everyone. This is Dheeraj, and today I'm going to talk about how flow cytometry as a tool is empowering our lab's quest to understand immune responses, especially in context of infectious diseases like TB, SIV, and now COVID. So for those who have gone through the abstract for the talk, here it is again. For those who haven't, I'd like to focus on the key points where I have referred to flow cytometry as a Swiss Army knife, especially mentioning to us the sensitivity and scalability it brings down on research, and then how it helps us in understanding host immunology for the infectious diseases, and how that knowledge is used to advance like vaccine and therapeutic intervention studies. So as you all can see, this is a nice multicolored knife set, which is a very common tool in every household. So if someone talks about knife, this is what comes to everyone's mind. But there is another variant which I would have preferred more than those sets. So this is how we see flow cytometry in our research terms. I'll go over why. So when we talk about biological function, flow cytometry has actually given us capabilities to measure varied of biological parameters, which includes DNA, which can be used for complete DNA content for a cell versus complete ploidy analysis. And then we can also look at RNA with flow cytometry with the recent advances or with like fluorescent RNA probes. And that can also go up to very specific time-lapse differentiation between cells showing specific transcripts. And then, as usual, the long-term thing are the proteins, and which is strongly used in immunology research for immunophenotyping. Now, recently, we have developed very good probes in the technology for metabolites. So it really helps a lot, even in the metabolic states for cells. And as usual, when like measuring all these different parameters, we can look what are the different transcellular states. So while these are the diverse functions that can be used by flow cytometry, the next best thing is the sensitivity. So usually if you're looking at a specific molecule, we can measure up to differentiating picograms quantities of different molecules in a system. For a cell, we can easily differentiate a couple of modulation pertaining to like 100 molecules per cell. Although with proper standardization, we can go even lower than that. And lastly, the most important part is we can do all of this at a single cell resolution, which by other techniques is not that readily possible. Now, the last part which really helps our research is the scalability. So we recently got a Symphony A3 bioanalyzer, and after that, our scalability options were like we could measure 30 parameters per sample at the rate of 30 to 100 samples per hour. Why that was especially important for us is something that I am going to cover in my next slide. So prior to moving to that platform, this is what we were doing. We had like multiple different panels. So this is an example panel for me. And on a typical day when we have a time point, I have to make like 11 panels like this, which will spend I'll spend 2 to 3 hours doing that. Then another hour, correct, like checking everything is documented properly. Now with the new scalability option with the bioanalyzer that we got, all I have to do is to spend half an hour, make a sheet like this, and with the new HTS setup, we could do all this within like an hour. And running the sample is like 6 to 7 hours job on the operator side, depending on how the machine is performing. So that really saved a lot of our time on the documentation part and also on the processing part, which we can actually use to do good research. Now I'm going on to the next part of my talk where I'll be talking about how we are using these techniques to understand the host immunology of infectious diseases. So our prime research focus has always been tuberculosis. And the main challenge with this bug is its generation time. So we used to call it a smart bug, which grows so slow that modulating any of its function biologically takes much longer. So the generation time for tuberculosis is 12 to 24 hours. When we compare that to other common bacteria, like for E. coli, which is 20 minutes, when we do research, it translates into something which is going to be a couple of weeks from when we start the experiment and when we get the results. So ideally speaking, if we set up an experiment, plate the bacteria, we can basically go on a world tour and come back and still have enough time to rest and then get to the results. While this is a key and crucial issue with studying tuberculosis, the main thing is when we are doing our standardizations, we really need to put in a lot of time to get to these kinds of results. So every time you are putting up a standardization experiment, you need to wait 3 to 4 weeks to get the end results to start on the next batch of experiments. So just to emphasize on that point, this is a sample vaccine test that we did last year. And it was a simple experiment where we were testing a candidate vaccine, which is the Delta SIGH, which is a SIGH deletion mutant. And we compared it to a BCG shown in green. And there was a naive control group, which was just infected, not vaccinated at all. And then after a while of vaccination, we infected them with a violent strain of MTV-CDC551. And throughout like a regular course interval, we did the blood and bile collection. And towards the end point, we did the necropsy for the animals. Now, what I specifically want to focus on this work is the timeline for this simple experiment. So if we call the day when we vaccinated the animals as day 0, so we infected them with the virulence from after like 2 months, which is a short duration compared to other models where we do long duration 6-month studies. But for this study, I was trying to focus on a small study so that I can give an idea of the timeline. So if we infect them at 2 months post-vaccination, we monitor the animals for like 10 to 12 weeks. And we finalize our last batch of experiments at around week 20 or 22. And after that, we still need a couple of like weeks to get the final end results. And a simple experiment like this in tuberculosis vaccine research take up to like 26 to 28 weeks. And it's like finishing the results, compiling everything is like 7 to 8 months of work. This is one of the key reasons that progression in TB vaccine studies is comparatively slower than other studies. But again, so this was an example where I'd like to talk about, do we really need to wait for like 7 months to get the final results? So here is a panel which we studied after vaccinating the animals and after infection, which was close to around week 12. And in the top left panel, what I'm showing you is a combined data from different animals. And the ones in the blue are showing the CD4 and the ones in the red are the CD8s. So based on this panel, we can clearly see that the vaccine is actually inducing a stronger effector response in the CD8s and a stronger immune response in the CD4s. And then we can look at the vaccine groups compared to the infected groups, where we can see, oh, there's comparatively slightly higher like effector responses in the early phase of infection. And on the right side panel, I show the different cytokines. So we can also tell that, okay, the T-cell response being generated are multifunctional in the early phases. And then on the same day, we can also do clustered analysis to see what kind of cell types are in there, what we are doing, and what we are getting in terms of immune responses. Now, while we can generate this kind of data by week 12, we can predict that based on this data, there clearly is significantly stronger immune responses in our candidate vaccine, both in the CD4 and CD8 compartments, compared to the unvaccinated or the BCG vaccinated animals. So even though we are not at the 7 months timeline yet, we can predict that how the experiment is working, and we can start planning on the next batch of experiments. And this is an unpublished data from the end of the study, where we can actually see that our immune responses generated by the vaccine were actually more protective in that group compared to other controls and compared to BCG as well. So while we are getting the other set of data in a couple of like 10 to 12 weeks, we really don't have to wait for like 7 or 8 months to finish the study and decide whether we need to move forward with the next studies, because this technique actually gives us pretty strong predictive capabilities so that we can plan ahead for all our experiments. Now, the next part, which is the main thing, is like flow cytometry cannot do everything. So can we flow through the tissue architecture? So while flow cytometry's main limitation is that it can be done on single cells, this is a set of H&E slides from the same batch of study that I just mentioned. And on the top left is the infected group, which has a large necrotizing granulomas, which are classic hallmarks of active TB disease. The BCG has comparatively smaller granulomas, and on the right side, the Sigurd deletion vaccine actually showed regressed granulomas, and along with strong induced BAL structures, which are lymphoid structures in the lung, and they're well correlated with protection from TB. So no, we cannot, like answering my own question, we cannot flow through the tissue architecture. But based on our expertise and based on this technique, we can predict how and what we are going to expect in the tissue architecture. And based on this, you can see that since there's a better T cell responses, we can expect the animals to be more protected from TB in the long run. Also emphasizing on the same point, we still don't have the master equation, right? So while we can do that with flow cytometry and while we can look at different parameters from different techniques using multicolor flow or like multicolor confocal as well, the main limitation about different techniques is that not everything can be applied to everything. So what we can see with flow is single cells in the periphery or like single cells arising from the tissues. But we cannot get a detail like this with flow. While we can do the same using other techniques like multicolor confocal. And to emphasize on that, I'd like to point out that this is a classical granuloma from a TB animal, and it has been stained for different color kind of parameters. But what I want to specifically point out in this slide is flow cannot show us this architecture. Flow can give us quantitative data. Now, my colleague, Dr. Bindu Singh, who is another postdoc in our lab, generated this image, and she also generated the quantitative data out of this, counting them manually for like a couple of weeks. The reason for that was some of the cell types actually give strong autofluorescence signal in these kind of tissues. And this was a stack generated from, I think, 16 different slides, or 16 different slides stacked together and stitched together. So while we were doing automated analysis on those were taking non-specific cells as well and RBCs. But do we really want to spend a couple of weeks on just single images? That's really not a good question. So what flow can tell us is what is the quantitative data and what we're expecting in the tissues, but we can validate that thing with other techniques, and we can go to exact architecture for the tissues in context of any disease. Now, while we are working on our TB work, we face the COVID challenge about which Deepak already talked about. So here's a sample slide from that study. And we generated some data in a very strong, like a very early timeline. And we could see that, okay, with the COVID challenge in rhesus macaques, we can see that there's a higher number of myeloid cells, including alveolar max, interstitial max, and there's a reduction in alveolar max in the BAL space in primates. And then again, we generated some flow cytometry data showing different dynamics in the T cells. While we had this data, we really didn't know anything about the virus, how it's infecting, or like how is it going to behave in our animal model. But we had a strong data of the cellular dynamics in the system, in the host model. And this data was generated within the first 2 weeks of the experiments starting. And then later on towards the end of study, when we got the viral titers, we could actually correlate the same data with the viral titers, showing that, okay, there is a very specific immune response being generated, and that is directly correlating with the viral titer. Just to go through the slides, on the top left is what where we are showing the complete viral load in terms of viral RNA. Genomic RNA, and on the lower end, we are showing subgenomic RNA, which shows that the virus is in a replicative form. So based on these studies, we could see that in the primate model, when we infect them, we could see a very high viral titers at day 3, while by day 14, the same viral titers are completely gone. But we could still detect viral load in terms of RNA copies if we are not looking to the subgenomic part. So they are not replicating, but there's still viral genome in the tissues. And we can also correlate that with the tissue, the cellular types that are infiltrating the tissues. So on our study, what we found out was that the alveolar max were negatively correlating with the viral loads, while the interstitial max were directly correlating with the viral loads. Other than that, we could see a specific kind of trend in terms of CXCR3 cells, both in the CD4 and CD8 compartments, and then a specific trend for CD4 and CD8 compartment PD-1 positive cells. So towards the end of the study, we could actually correlate and understand how the immune responses are getting generated with the virus or the bacteria, and then we can correlate that with how it's changing with the disease dynamics. Now, since we had that data in right time, the next question we can ask is like, are we looking at the right place? So we had myeloid cells increasing, but are they actually harboring the virus? And this is another very nice slide from one of my colleague, Dr. Bindu Singh, where she actually showed that, yes, this myeloid population are actually harboring viral proteins. And just to summarize everything, I would like to conclude by saying that, yes, we can accept the challenge by using different techniques and bringing them to the same platform to understand whatever challenges we are facing in infectious disease research. Towards the end of my slide, I'd like to thank Dr. Deepak Kaushal and all the current lab members of the Kaushal Lab. I especially like to thank Dr. Tae-Hyung Lee, who has been our resident expert in flow cytometry. And then lastly, Dr. Bindu Singh, who is our resident expert on confocal microscopy. And most of the generator presented in these slides have been generated by me with their assistance. Thank you. Towards the end of my slide, I'll just leave everyone to ponder on the laws of cytometry. I specifically put this slide because I have met a lot of people who really don't know that there is a thing which is called as the law of cytometry. So I was just like to point out that, yes, there is something that is called the laws of cytometry, and it exists from even before I was born. So I recommend everyone to read a very elegant book by Dr. Howard Shapiro, which was published as early as 1985. Thank you.
Well, thank you, everyone. And I just really like to take this opportunity once again to thank Dr. Kaushal and Dr. Singh for just beautifully presented, studies. I think before we get to the questions, though, I just like to say that it's stunningly beautiful how your team has been able to transition your work so quickly from TB to COVID projects. The amount of time and effort and the skills that are necessary for that are I'm sure are just very, very big. And for your teams that have worked so hard to transition through all of that work, I want to say also for all of us out here, thank you very much. Today, though, we have a whole series of questions, and I hope we can get through a couple of them because we're coming up to the top of our hour. But Dr. Kaushal, for the first question for you, the data on the vaccine indicates that by day 9, there is an attenuation of immune responses. Are we considering ways to extend the response, and in that, the reason why we need to have 2 doses of the vaccine in order for it to be effective? If you could comment on both those.
Yeah. So yes, these things are being considered. Not just our group, Dheeraj and I, but and so many others at Texas Biomed, but also other investigators outside of us are considering these approaches. One such approach is to use comorbid animals that have defined cardiovascular disease, in addition to just being aged, or type 2 diabetes. There are other molecular tricks that one can potentially use, but all of these experiments are expensive and we need more money and more funding. One way is to potentially take out protective cytokine signaling, such as type 1 interferons. So Dheeraj and I have some experience in doing this in non-human primates in the context of TB, and we can do that in SARS-CoV-2 infection as well. So you can imagine if you blockade type 1 interferon signaling, that maybe you will get more exacerbated disease. So there are some ways of doing it. It's just that we need more funding and we need more support from all aspects to be able to do these incredibly complex and expensive experiments. And then the second part of the question, Bob, was why do we need 2 shots?
For two doses.
Why do we need 2 doses of the vaccine? I think that's your classical amplification of the immune responses. So, we're not just looking at early ablation of the virus by myeloid cells, but proper T cell responses and antibody responses, just an amplification effect.
Oh, very good. Thank you very much. The second question for Dr. Singh, the innate lymphoid cell 3, the ILC3s, has shown to play a role in TB. Do we suspect that they may also play a role in the SARS-CoV-2 infection?
So, yeah, because of the initial workload, what we were initially focusing on was the adaptive immunity. But later on, some other of my colleagues took over the work, and they also analyzed the samples from the same study sets. And Dr. Tae-Hyung Lee actually had very strong data, which shows that in the initial phase of the disease, the NK cells, although they start proliferating, their effector responses are completely diminished, and they are blocked in some way to get the effect on the virus. And that happens until day 6, which was our time point. So we can see that on day 3. We have seen that in day 6. And then after they just start gaining their function with the resolution of the disease. So yeah, the functions of NK cells actually correlates well with the disease. So when the virus is going up, their function is blocked. And when they start regaining their functions, the virus is going down. But how much of it is actually caused by the virus, and how much of it is actually caused by the NK cells is something that we need to study in a more controlled setting. Other than that, we could see that the good responses were more pronounced in the young ones compared to the old animals. So that can be another reason why the disease manifests in a different way in these animals. And one final very strong data was that young animals had a very strong antibody responses. And one of the key mechanisms for NK cells to exert the effector function is through antibody-mediated cell cytotoxicity. So that's another direction that we are moving on with currently. So there are a bunch of studies going on in different directions by different very talented people in our group. So yeah, we'll get back with those data soon.
Okay. All right. Well, I thank you both very much. I know that we're at the top of our hour. I want to thank all the participants that stayed with us through the day. Once again, thank you both very much for the presentations. I'd also like to thank my team too for all the work they've done. Thank you very much.
Bob and Manisha, thank you for having us. It was a great pleasure. Thank you.
And thank BD, the entire BD team, for the great support they have on immunology research.
Thank you.
Thank you.